So we are recording now, and thank you very much, Vincent, for agreeing to give the Build a Cell seminar today, and it's all yours.
Thank you, Kate. It's a pleasure to be here. I'm going to give a presentation on cell-free expression and how we use it to construct biochemical systems in vitro. And I will pass relatively quickly on the first items to focus more on the synthetic cell or minimal cell systems. You see some images at the bottom here. We develop a self-free expression system from E. coli that allows us to construct biochemical system in vitro using regulatory elements from E. coli and from bacteria. CRISPR works very well. We're able to express a large gene sets such as phages that are infectious when they are produced in vitro. And I will focus today towards the end of the talk more about the synthetic cell system. You see here some liposomes that were spherical at the beginning, and they take the shape of rods when we express a cytoskeletal protein. But the most important here that it's entropically driven. You need molecular crowding. So a snapshot of the lab activity at UMN here. We use self-free expression in test tubes or on well plates, if you like, and we put the self-free reaction also in liposomes to make synthetic cells. And the idea is to construct a biological system outside the living organism using DNA programs. I don't want to purify proteins. I want to do like a real cell to express genes. We have done a lot of prototyping. The last one is on CRISPR. We have done, we demonstrated oscillators. Right now, the lab is focusing more on synthetic life, on two parts, synthetic cells and phages. And we are trying to develop also biotechnology applications. I'm going to skip those biotechnology applications today and focus more on synthetic life. Before giving an introduction to self-free expression systems and what's different with the E. coli self-free system that my lab has developed, here is the quick outline. I give an introduction on TXTL, the E. coli TXTL, TXTL from other bacteria, something new that I have done with Wang at Columbia. I will pass very quickly on prototyping, taking two examples very far, very simple regulatory element, CRISPR, and very large gene sets with phages. And finally, I will focus in the last part of the talk about synthetic cells. The work that I'm going to show has been done by those postdocs and graduate students and undergraduate students, and the funding is acknowledged there. That's it. So let me introduce TXTL. What I say with TXTL, TX is for transcription, TL is for translation. It's self-free transcription translation. There are other words for this like CFE, self-free expression, and IVTT, in vitro transcription translation. Just use TXTL. It's as practical, I would say. So self-free synthetic biology, the major goal is to construct biochemical system in test tube using DNA programs like you see with the image in the center here. And there are many advantages with that and also disadvantages. So the advantage is that one of the major advantages that it's bottom-up, reductionist, and constructive. It's a TXTL system is fast, it is safe. As you know, synthetic biology is literally, I mean, right now growing up like crazy. And what I like with self-expression is that it's very safe. And what I like also is that it's quantitative. In everything that you are going to see, there is no endogenous information. So the information, the DNA that we add to the self-reaction is the one that we clone in the lab. Or we use natural DNA like phages. You will see this on one slide. What I like is that there are no interference and response from an organism, so there are more freedom of control and design compared to in vivo. And the demonstration of the practicality of self-free system has been demonstrated many times now. It's really a useful platform to accelerate synthetic biology, especially prototyping and biomanufacturing. I was saying that there are disadvantages. It's self-free expression is not as powerful as gene expression in a living organism, but you will see the last cell-free systems are powerful enough to really execute large DNA programs. Quick overview here of TXTL. You have to choose an organism. E. coli is still mostly used here, but many labs have demonstrated that you can use other organisms. You have to grow them in vessels like this, in flask, and then you have to prepare a lysate. You have to break the cells, and then there are different steps to clean up the extract, to remove the genomic DNA, the RNA, the messenger RNA, and so on, and to keep especially the translation machinery. And after this, the last step here, you have to compose some buffers, some amino acid mix, some energy resources, and then you clone your genes, and you can express genes like this, either in a test tube, on well plates, or in large vessels. Some characteristic here, the reaction volumes of self-reactions is just phenomenal. You go from the femtoliters, the size of E. coli, up to some liters, and there are some companies doing 100-liter self-reactions. Flask, well plates, so flask for large volume, well plates on the scale of a few microliters. Microfluidics, very popular. At that scale here, a fraction of nanoliters, and finally synthetic cells. You can go down to femtoliters. You will see this a little bit at the end of the talk. So another overview of cell-free SynBio applications. Very popular is prototyping, and it's still done extensively. You can very quickly get the performance or the characteristic of regulatory elements, of circuits, of interactions between proteins or proteins with DNA, of pathways, sensors, and so on. Biomanufacturing is also developing very quickly right now. You can use a self-expression system to prototype, for example, antibodies. And very also quickly developing here are diagnostics. Especially the paper-based diagnostics have been demonstrated in the past 5 years, 5-6 years. And finally, synthetic life, it's picking up very well all over the place, around the world, in Asia, Europe, and now in the US. It has become a sound objective to try to construct living cells from self-free expression system. It's actually, I consider that the self-expression system is a major route towards building minimal cells right now. So that's it for this first part, a quick introduction to self-free expression. What my lab has done on this is to develop what I call the whole E. coli TXTL. And a major difference here that we use the E. coli RNA polymerase, the core RNA polymerase, and the sigma factor 70, which is a primary sigma factor in E. coli, to boot up, or to start, if you like, all the gene expression in the system, mostly like E. coli. It's very different, as most of the self-expression system at that time when we started to develop this platform were based on the T7 RNA polymerase and the T7 promoter. That's it. So here it is, a quick also overview. It's like this in a test tube or a well plate, or you will see in a liposome. You can use linear DNA, PCR product, or plasmids. And once again, you will see the major differences now we use for transcription, the core RNA polymerase and sigma factor 70, and that gives you hundreds of regulatory elements now that you can use from E. coli or other bacteria. You are not limited to just the T7 promoter and the T7 RNA polymerase. The lysate is prepared, as we have shown many times in articles, from the E. coli BL21 strain, Rosetta 2. It is stable for many years at -80. We were recently still using some lysate from 2015, and they work still very well. The energy is based on ATP regeneration and glycolysis. We have a new system coming up that we hope to publish 6 months earlier, the toolbox 3. You will see a few characteristics in a few slides. We have a mix of 20 amino acids, and the 20 canonical amino acids. There's nothing special there. The lysate contains all the tRNA ligase. There are no post-translation modification in this system. It's one of the characteristic and maybe disadvantage, but still so many proteins can be expressed the library is huge here. And measurement, you will see we use reporter proteins typically, EGFP, but the other reporter proteins that work, ECFP, EYFP, Venus, MreB, and so on. So here it is, I insist on this. We start with promoters, regulatory elements specific to this transcription machinery, the E. coli core RNA polymerase sigma factor 70. You see GFP after a few hours. We are even more powerful now. So we express up to 4 mg per ml in batch mode. So we have reached 150, 160 micromolar of GFP in batch mode without feeding the system. The kinetics, they look like this. The dark is plasmid, the open circles here are PCR. This is EGFP in micromolar at the time. So you accumulate the protein and then you reach a plateau where everything has been consumed. It's a chemical equilibrium. There is nothing happening anymore, just stops here. And so we demonstrated that you can recapitulate the entire sigma factor transcription scheme from E. coli. All of the sigma factor, they can be expressed and they are specific to their promoters. I'm going to take the example of sigma 28. You use it like this. From a P70 promoter, you express the sigma factor that recognizes a specific promoter to express GFP, and you get the same kinetics. I just show here the plasmid expression. This is again GFP and the time. You have a window of 8 to 10 hours to express protein. And all the other sigma factors, and including the T7 RNA polymerase, the T3 RNA polymerase, you can use them using this circuit scheme here by just cascading. That's it. The toolbox 3 is coming up. The toolbox 1 was done with my first PhD student, Jongyang Chin, 2012. The toolbox 2 was done with 3 graduate students, Ryan Marshall, John Garamella, and Mark Rostad, published in 2016. We hope to publish the toolbox 3 in 6 months or a year. Let me give you 2 characteristics. It was a toolbox 1. We were at barely 1 milligram per ml of GFP in 2012. We are a little bit more than 2 mg per ml in 2016, and we have reached 4 mg per ml. It's batch mode, it's not continuous mode. 4 mg per ml in the past few months. It's one of the results in the toolbox 3. Another results here, it's just, you see, just the expression of GFP. What we do also, we test the toolbox systematically with a very large DNA program, and our test is a T7 phage. So you can literally buy the T7 genome. And we use it like this. We put that in the self-reaction and we look at the number of phages that are produced. It's called PFUs, plaque forming units. It's literally the number of phages that you count with a plaque assay. And here are the synthesis of the phage T7 over the years. In 2012, we were at 10 to the 9. We start at 10 to the 8 here. At 10 to the 9, the toolbox 2, we are at 10 to the 11. And from the toolbox 2 to the 3, we have a factor of 1,000 here. With the new system, we have reached 10 to the 14 phages T7 produced in a batch mode self-reaction. So that's very large. And again, we have other results here. We hope to publish this within the next year. Let's put it this way. So before going to quickly some prototyping example and the major part here, the synthetic cell, I want to show what we have done recently with Wang at Columbia University. We showed that you can produce TXTL from other bacteria. They are far less efficient than E. coli, but still working. And you will see that using next generation sequencing, you can start to do really massive prototyping of regulatory elements. I will show you promoters, how we characterize promoter using NGS here. So what we have done mostly in the lab of Aris at Columbia University, done this cell-free expression system here. There are about 10 there from gram-positive, gram-negative bacteria. You have proteobacteria, actinobacteria, and firmicutes, including very, very well-known established organisms like Bisoptilis and Vibrio natrigens. Some other people have done recently some cell-free system from those organisms. And we use this as once again, a very powerful, massive, parallel prototyping. Those cell-free systems, they are prepared the same way as E. coli. You grow them in flask. You do a lysis. Here we used sonication. Then you clean up the lysate, and then it's ready. You store it, and you can do massive prototyping. What we do there, we use the broccoli. We use a fluorescent aptamer to look at the performance of promoters. And what we did, we used next generation sequencing and barcoding. We barcoded all of those regulatory elements. Each time you have a different promoter, and you are going to look at the production of an aptamer. Or literally the production, the aptamer was used to define the biochemical setting of the lysate with respect to magnesium, potassium, and so on. And after you use barcoded DNA with promoters to and next generation sequencing, and you look at you count literally by next generation sequencing the frequency of your RNA that is produced from a library of promoters. Here it is. Let me show you just this example. You can go up to 1,000 promoters in just one tube. That's it. Those hundreds or up to 1,000 different regulatory elements, and here they are promoters. And you use next generation sequencing to look at the frequency and the strengths of these promoters, and you can align everything by sequence analogies here, from very, very strong here up to weak promoters. And you can start to do a lot of statistic work and compare the strengths of the promoters just in one test tube reaction. You see they are annotated on top. It's very small. We have 400 in this example, and that's pushing the prototyping approach using TXTL to another level, and that was done again with the laboratory of Wang. Let me return to the prototyping more standard style, and I'm going to take two examples here. Very far prototyping CRISPR technologies and prototyping executing large gene sets. What you can do right now in terms of prototyping, here it is. I showed promoters, but you can do UTRs, RBS, terminators, any RNA aptamers, riboregulators, transcription factor, any genes. And the last one that we have done in collaboration with the laboratory of Chase Beisel now in Germany, is do CRISPR. And you can also prototype gene circuits. I will take the example of a phage just to show an example of huge, large gene sets. That's it. So CRISPR, just very quickly, you literally put in your cell fraction a plasmid to express the CRISPR enzyme, like DCAS9. You put a guide RNA and a target gene, which is GFP. You can use DCAS9 or Cas9, and then you have a targeting where you are going to repress, and you have a non-targeting, and you can determine the on-off ratio of any guide RNA at any place on the target gene. So guide RNA works very well. We have now, we are also writing papers on CRISPR activation. We have shown CRISPR inhibition or interference. You have worked with James Carothers at UW here to show that CRISPR activation works very well. CRISPR enzyme, almost so far, all the CRISPR enzymes that we tested, they work. Anti-CRISPR protein, they work very well. And we can also do PAM assays. That's it. So everything works very well with CRISPR technology in TXTL. Let me take a very far example. We went from prototyping or looking at the characteristic of a regulatory element like a guide RNA or a CRISPR enzyme. I'm going now to take very large gene sets. Those are the 4 phages that we have synthesized just in test tube like this, and all of them are infectious. They have the same characteristic as if they were expressed or synthesized in E. coli. So MS2 is an RNA phage. It's very small. 5X174 was, I think, the first phage discovered. It's the size of a plasmid, 11 genes. We have T7 that we published in 2012 that start to be bigger, 40 kilobase per 60 genes. I like this image because what you see here is a cell-free reaction without cleaning. So you see the phage particles and all the small bullet. You see the small bullet all over the place. They are the ribosomes. And you can see the phage packaging here and here. And the last one that we have done in 2018 is to show that the T4 phage, which is 169 kilobase pairs and almost 300 genes, it is fully synthesized in a cell-free reaction. It is currently the largest DNA program that has been expressed in a cell-free reaction to give a functional unit here, which is a T4 phage. All right, let me conclude the talk. We have 5-10 minutes on synthetic cells. Everything that you have seen is done in test tube or on well plates. Now we are going to encapsulate the cell-free reaction into liposomes. They are phospholipid vesicles. The physical boundary here, it's a phospholipid bilayer. It's typically from 1 to 50 micrometers. 50 micrometers you can get sometimes, but they are not that stable. It's typically from 1 to 15 or 20 micrometers. And you can do exactly the same as in a test tube. You just you have to use a method to encapsulate the reaction, and then you will see GFP expressed after a few hours. Before going to this, to show some expression in liposomes, let me cast a little bit the landscape here. There are different type of bottom-up synthetic cells. We do one type in my lab so far, a little bit of a second type. I will not talk about this in the talk. You see there are a lot of words here: artificial cell, minimal cell, protocell, semi or synthetic cell, reduced cell, coacervate, and so on. It's a lot of words. You can encapsulate this into 3 categories. The first one are protocells. When you do protocells, you look at the origin of life, and in particular, the origin of unicellular life. And it means that you use peptides, RNA, and fatty acid. You use prebiotic, or what we think are the prebiotic molecules. And you do protocells. Kate knows that very well. She has done this during a PhD with Jack Shostak. Artificial cell, artificial by definition, it's when you use a component that is not natural, that is artificial. So you synthesize a cell size compartment that incorporate artificial molecules. A number of groups are doing this. And finally, what is called the minimal cell, it's what many labs do. You try to synthesize what is called a minimal cells, looking towards self-reproduction using the natural molecular machineries or the natural molecules, if you like, typically phospholipids, a cell-free transcription translation system, genes, and so on. There is nothing synthetic there. Everything, or if you like, the building blocks of the chemicals here are natural as opposed to artificial. And so let's go to the minimal cell now. So what we do, one way to look at this is when you want to make to build a minimal cell from the bottom up, you have to think about integrating 3 molecular blocks here: information, gene circuits, if you like, compartment, or I extend compartment to self-assembly. You have to consider that molecule is self-assembled, and then metabolism, and you shoot towards self-reproduction. So each of those parts here, they are essential. If you remove one, that cannot work. We think that removing information at the beginning, millions or billions of years ago, just with compartment and metabolism, a possibility that the first cell, they emerge just with compartment and metabolism. It's just there's no proof of that. It's just an idea. But right now, each part is essential. You need information, compartment, metabolism, and each part is made of molecular machineries. And so what are the objectives? It's just, the far goal is self-reproduction. It's very ambitious, of course. You want to capture the cooperativity between those blocks here. How does that work? How you have to integrate those blocks so that you start making something that works. And you look towards self-reproduction. Meanwhile, and everybody agrees on that, you can construct minimal cells that are very specific to a biological function to develop specific applications. And there are a growing interest in doing this, in trying to integrate one or two specific functions for applications. So here is a TXTL bottom-up minimal cell. I took this image from this article from Pasquale Stano and Pierluigi Luigi. There are other articles that I have written also on this. You try to encapsulate literally your self-reaction with your DNA and the nutrients, and to express genes in cell-size liposome using the natural molecules and building blocks, and try to start recapitulating biological function. I'm going to focus on two examples here, what I have done with Alain Liu these past few years. It's a kind of signaling system, And I will take another example, what I have done with my postdoc, David Garin, about molecular coding. We determined that molecular coding located at the membrane is critical to self-assemble cytoskeletal molecule. Let me start with what I have done with Alain. Alain is at the University of Michigan. We use the emulsion method. So you make an emulsion. Here is your reaction, like in the test tube, but now you make an emulsion. It's in oil. And you have a first layer of lipids assembling around the droplet here, and you pass this through an interface to make the second leaflet here and to make your liposomes that are transferred into what we call a feeding solution or a buffer, if you like. And what we developed in the past few years is some signaling system. We are going to express, you see, I'm using the Sigma 28 cascade here to express MSCL, which is a mechanosensitive channel. It responds to the tension in the membrane, opens up, and then leak molecules to balance the osmotic pressure. So here it is. What you see is one liposome here. The red that you see here is just a dye. We put a dye of a few kilodalton in size. It's labeled with TRIC, and when you express something else than MSCL, the dye you see over time stays inside. We are going to do exactly the same, but express MSCL and apply a tension, just an osmotic stress. And you see that relatively quickly, MSCL is expressed here, it's not labeled, but the dye leaks outside the solution here that is around the liposome, that's it. So we build up something a little bit more sophisticated based on this result. Here it is. I'm going to start here on the left. You have a switch here. You have a switch. We express the lac repressor to turn off the expression of MreB, a cytoskeletal protein, and we express constitutively MSCL. So MSCL is going to go to the membrane. And what we do, we put IPTG outside, and we apply a tension. We do a plus-minus tension to see if MSCL opens correctly, the IPTG leaks inside. We open that switch and express this cytoskeletal protein. Here it is. It's at time 0, and you see when you are in hypoosmotic conditions, we effectively open up the membrane, diffuse the IPTG inside, inhibit that switch here so that we can express MreB that co-localize at the membrane and make the liposome tougher in terms of mechanical robustness. That is negative control where no MSCL is included here as a plasmid in the liposomes, and that is a positive control when we put IPTG right away inside. It's a little bit brighter in that case, but we were able to show that statistically what we show here on this picture is relevant. That's it. I'm going to conclude the talk with that last result. I'll come back to MreB. It's something that we have understood only in the past 10 months, I would say 10, 12 months. So here it is. MreB is an analog of actin in mammalian cells, and it forms a structure at the membrane of E. coli for mechanical stiffness of the bacteria. I mean, that has been shown. E. coli has a peptidoglycan, but it's not enough. MreB is very important for the mechanical stiffness of E. coli. And it not only that, but it gives the shape of E. coli. If you remove MreB, you get some E. coli cells that are spherical. MreB gives the rod-like shape. That is E. coli that you see. You see it makes some kind of structure, helical structure in the bacteria. So here is an image where you do cell-free expression. After a few hours, we express GFP there. And here is when you express MreB that is fused with the GFP. And if you express MreB, you're not going to see this. You need something else. So let me explain to you. I just 2 or 3 minutes here and I'm done with the talk. Let's start there. So here is a liposome. You express MreB, any GFP MreB. There is a small amount of GFP MreB, a ratio factor of 10 here. If you put 1 nanomolar of MreB here, you put 0.1 nanomolar of GFP MreB, you put the TXTL. And here is a membrane made of phosphatidylcholine only. You can see the lipids there. And you do exactly the same here, but you change one thing. You put some lipid that has a PEG attached to it. So on the head here of the lipid, there is a PEG molecule. And this PEG molecule is going to do some molecular coding responsible for assembling and activating MreB as a cytoskeleton. Here it is. So MreB is expressed. You see, it goes to the membrane. It colocalizes with the membrane, but it stays spherical. We don't see, we barely see some deformation. And you need this molecular coding to change everything. Now, MreB goes at the membrane, self-assemble, makes filament. The persistence length of MreB is about 5 to 10 micrometers. It means that below 10 micrometers, it's a rigid filament, and above 10 micrometers, it is soft. And you see that it deforms essentially almost all of the liposomes. We made some statistical analysis. We bin the liposome by size. I'm going to show you this here. So the liposome are binned by size from, you see, 0.2 to 4 and so on micrometers. And we look at the percentage of the liposomes here that are in those long over short axis. If you take this liposome, this is a long axis, and the short axis is like this. We measure this for a population of liposomes and did the statistics. No deformation when there is no molecular coding at the membrane, and deformations, especially at small size here. And that correlates very well with the persistence length of the filament here. It's about 5 to 10 micrometers. And so below, you see below 5 micrometers, we see a lot of deformation, and above, the deformation are much less abundant here. What we have done, one of the questions, let me explain to you that just quickly, an explanation. It's driven by entropy. So let's take some molecules here. You see the gray ones and the dark one. The molecule is a gray one here, and you see a circle here around. It's the excluded volume. If you take another molecule, you see the center of mass. I am just at the center of mass of 1. You cannot put it in this white region here. You cannot physically put it there. And you ask the question, how can I put one other molecule there? You can put that molecule, the center of mass, in the right space here. You see, you have a little bit of right space, but not that much. And so those gray molecules that do not do anything, they are going to, the dark one, let me be more clear here, the dark one, they can assemble together. They are the MreB ones. Those gray molecules, they are going to push and to promote the self-assembly of the dark one because the entropy increases. So now you ask the same question, how can I put one molecule once I have self-assembly? This is exactly the same number of gray particles that are shown. You see that after self-assembling the dark ones, well, you have much more space now to put another one. The conclusion is that the free volume created due to the self-assembly is much larger in that case than in this case. So it's absolutely entropically driven. So let me conclude with that slide here. One of the questions that we had is that, is it depending on the type of molecule we use for molecular coding? And at the beginning, we used PEG. And we asked the question, if we put something else than PEG at the membrane, does that work? And the answer is yes. I'm going to skip this image, it's just reconstitution here. I'm going to just start there. Now, this is what we were doing before with PEG. That is the same type of system. Now we use we use streptavidin biotin. We use streptavidin biotin, and we put some streptavidin inside, and because we have lipid biotin, the streptavidin stick to the membrane and form a molecular crowding that has nothing to do in terms of chemistry, nothing to do with the PEG. There are absolutely two different types of macromolecules that we stick to the membrane. And the fact is that it works as well. When you put streptavidin, you deform the liposomes, you force the self-assembly of MreB, and you get a statistic that is comparable. We do the same long over short axis statistical analysis. We bind the liposomes, and you see that it's absolutely comparable. We get the same thing. So the conclusion is that you need a macromolecular background at the membrane to accelerate, to promote the self-assembly of cytoskeletal protein at the membrane and make them active so that they, you go from spherical liposome to rods that looks like E. coli. This is the last slide. I want to advertise this course here. This is a Cold Spring Harbor synthetic biology course. Of course, as you know, with no surprise here, it was canceled this year. We were supposed to be there in just a few days. But we hope to be back next year. I just want to advertise this. It's a 2 weeks long course. It is very intense at the bench. We do experiment every day. We invite a lot of speakers. We have a speaker almost every day. And if you are interested, if you are a graduate student, a postdoc, or a faculty, we have typically 1 or 2 faculty in the group every year. Please do not hesitate to apply or to ask us questions, to ask me questions about this. And that's it. That was my last slide. I will stop sharing.
Thank you very much, Vincent. That was an awesome presentation, as usual. We do questions mostly through chat. If someone wants to unmute themselves and ask, feel free. But you already have first question in chat, Vincent.
Has TXTL been tried yet for streptomyces? Not that I know. I have not seen this. But I think some group are trying. I think the group of Mike Jewett or there was another group in the UK. I think they were trying. So I don't know if it's published yet. They were trying for some metabolic engineering purposes, I think. In E. coli, what recruits MreB to membrane and what holds it there? So MreB, it's an analog of actin, and the N-terminal part of MreB, the first 10 to 20 amino acids, they naturally interact with phospholipid membrane. I forgot to mention this. So MreB spontaneously stick to the membrane. And that's why it is found at the membrane. Yeah. Is the explanation of molecular crowding valid for cell? I suspect, yes, I suspect that molecular crowding explains not everything about MreB self-assembly, but it explains the acceleration of the self-assembly into filament. It's what it explains. And so your question, the follow-up question about the crowding in terms of percent of the array, it's a very good question. We made a study of this in our article that we published. You need to be relatively precise about the concentration of lipid PEG that you use. And it also depends on the size of the PEG that you use. Typically, we use a lipid PEG with a PEG of 5,000 in size, 5,000 grams per mole. We show that it works also with a lipid PEG 2,000, lipid PEG 1,000. After at 500 and below 500, it's harder. It works not as well. So let's take a PEG, a P-PEG 5,000. An optimum concentration is around 0.66 to 1% in the membrane. And we explain why. Above 1%, the PEG is going to cover the entire membrane. and you start screening the membrane so that MreB cannot bind to the membrane because you have a shield of PEG molecules. That's it. That was the question from Bhagyashi Shri Mahajan. The question from Mark Frieding. You mentioned that you get a yield of 4 micromolar using TXTL. How does this compare of expressing an insoluble protein like MSCL? What is a typical yield? We have expressed only EGFP so far. We have not measured the expression with a new system, with a toolbox 3. We have not measured the expression of membrane proteins yet. But that's a good question. It's something that probably we want to do in the next few months to get a sense of that. Absolutely, yeah. So not done yet. Question from Amir. Pendy, have you seen different behaviors with transcriptional activators versus repressors? That's a rather broad question. They all have different characteristics. Transcriptional activators, let's take the sigma factors, they work absolutely differently. Some transcriptional activation factors, let's say sigma factors, are very powerful. And some others are not as powerful as sigma 70. Let me give you an example. Sigma 28 is very powerful, potentially as powerful as sigma 70. Sigma 19 and 25 reasonably powerful, but not as strong as sigma 28. And repressors, you know, repressors, we tested the very conventional repressors so far. We have lack, of course, TET, arabinos, we have melibiodes, we have, so far all the repressors that we tested, they work, including CRISPR interference. CRISPR works very well in TXTL. A question from Ali. I want to know you can handle glycolysis waste in self-association, especially in batch mode. This is a very good question. It's the major problem in batch mode. In batch mode, what turns off the reaction in batch mode, it is the accumulation of byproduct, or the waste, if you like. And it's a major factor that turns off the self-free reaction. So it's a question really of settings. It becomes a very fine mechanics here. Settings between the magnesium, the potassium, the sugar that you use, and we managed to go up to 4 mg per ml. Surprisingly, I was not expecting to go more of let's say 3 mg per ml, but we have reached 4 mg per ml, which is again much more than I could have imagined, in fact. For many of your constructs, why do you use a cascade sigma 70, sigma 28? That is also a very good question, a very good technical question. We use genes from bacteria and a lot from E. coli. And if you clone those genes under a promoter specific to sigma 70, they are going to leak in E. coli when you clone and when you prepare your plasmid. And very often they are toxic for E. coli. So we use a promoter and the cascade sigma 28 because it's a very silent promoter in E. coli. And that allows us to do our cloning of toxic genes. And we express those genes using the cascade, and that's it. It's just really a trick to clone and to express genes that otherwise are toxic for E. coli. Question from Christopher. In your figure, with the gray and black nodes in entrepreneur, what corresponds to the gray and black MreB? Sorry, I should have been more clear. The gray are the crowders, let's say the PEG, and the black, they are a MreB, the MreB protein, that's it. And so the black, they self-assemble, and the gray, they don't do anything. They do not interact with MreB, they don't do anything, they just sit there, but they promote the self-assembly of MreB, that's it. Other question here, can we express two proteins simultaneously with control stoichiometry ratio using TXTL? Absolutely, you can do that. You just tune this with a plasmid concentration. There are many proteins which needs, let's say, specific stabilizing protein around. You can do that. And we do that frequently. We express up to 5, 6, 10 proteins together, and we just change the concentration of the plasmid. That's it. If you have E. coli machinery, another question from Fernando here. Machinery in cell-free extract, the plasmid can replicate themselves? No, we have not. We do not replicate the plasmid. So let me develop a little bit more here. I think nobody has done plasmid replication so far, but one group, several groups have shown that linear DNA you can replicate it using the phage 529 DNA replication machinery. That has been published and demonstrated. So not for plasmid so far. I think it will be done at some point, but for linear DNA, you can replicate the template that you put in the cell-free reaction. Question from Stephen here. Does protein degradation remain functional? Are you able to express small unstructured peptide? So does protein degradation remain functional? Protein degradation using the ClipXP system works. It is functional. We demonstrated that in the toolbox 2, for example. So it works. You need to tag the protein with a degron, a tag that is specific to ClipXP, but that works well. Are you able to express small unstructured peptides? That's a good question. We have not expressed that many small peptides or proteins. I don't see why not, but that should be verified. That should be verified. So I don't have a sharp answer for this question. Question from Amir, if I am not mistaken, plasmid replication has been done in pure system recently. Exactly, it's what I was saying. So linear has been done, plasmid I think has been done also, but it's specific to the pure system. If linear DNA PCR fragment were, why still using plasmids? Very good question. It's because expression from a plasmid is stronger, is more powerful, more efficient than PCR fragment. That is due to a question of conformation of the DNA and stability of the DNA. In the system, in the E. coli TXTL system, plasmids are absolutely stable. PCR product, we found ways to stabilize them, but still it's not as the expression from PCR product is not as strong. That's why. That's why, and in my lab, we are doing both. We are using plasmids and PCR product. Question from Marco. Marco, thank you. Is there a defined limit of component in a genetic circuit expressed in TXTL or co-expression of many proteins in the same reaction? The only limit here, it is the capacity of the system, or if you like, the energy. At some point, you are going to reach the maximum capacity, and then you will start sharing the resources, sharing the ATP, GTP, sharing the amino acids. And so it really depends on the load, on the number of plasmids, the strength of your promoters, the strength of the RBS that you add to the system. That's it. And I think that's it. No more question. Anyway, it's almost time. Yeah. Yeah. I think, Kate, I think we are done essentially. Yeah.
Yeah, I think you answered all the questions. Thank you very much. I have a few more of my own, but I'll ask it offline because I think we're running against the time limit. Thank you very much.
No, thank you. Thank you. It was great. It was a great opportunity. Yeah.
Thanks, Vincent. And thank you, everyone else. And see you next week.
Thank you. Bye.
Bye.